Right. So previously we were talking about the process of transcription, which involves taking a section of a DNA and then making a copy of it in the form of a messenger RNA. Now, the messenger RNA that you're going to be getting is called pre-mRNA because it is still premature. It's not ready to be taken out into the cytoplasm just yet. Now, when you do the transcription, there are three steps. The first step is initiation, where the RNA polymerase is going to be binding to the promoter. Promoter marks the beginning of the gene, right? And it's going to unzip the DNA, and then it's going to start making copy of the DNA. Now, what's really confusing about this situation is DNA is double-stranded, but RNA is single-stranded. So you are not going to be able to copy both of the strands. We are only going to be copying one of the two strands. Okay. Of the two DNA strands, one of them contains the actual instructions to make the protein. We call that the coding strand. Okay. The coding strand has the code that would make the protein for you. And then the other strand is called the template strand. That's the strand that we'll be using to create the pre-mRNA. You cannot copy the coding strand directly because if you did, everything is going to be backwards. Okay? When you do the building using the polymerase, it's always going to be following the base pairing rule. Okay? If it sees a T, it's gonna build an A. If it sees an A, it's gonna build a U. Right? So that's why you have to be using the opposite strand so that when you create the pre-mRNA, it's gonna have the same information as the coding strand. And so on the test, on the quiz, I will tell you whether it's the coding strand or the template strand, and then you will have to work with it accordingly. Now we're gonna do some practice right now. Okay. 202, March 13. And let's just copy these things over. If you don't understand, you have to ask, okay? Otherwise, you're going to lose a lot of marks on the quiz and on the test. Example 1. Given the template strand, find the coding strand and the mRNA. Right? So if I just give you this strand and I don't say anything about it, you wouldn't know whether it's template or coding. But the question says it's template, right? So we're going to put template right here. And you are asked to find the coding strand. Remember, both template and coding are DNA. Right? DNA has two strands. Right? What's the relationship between the two strands? What's the word that we use to describe the relationship between the two? Yeah. Complementary, that's right. So if you have the template strand, you should have no problem creating the coding strand, right? Okay, so let's take a look at what the coding strand will be. If it's A on the template, what would it be on the coding? Continue. G, yeah, a little bit louder, don't be shy. Yeah. A and then? That's it. Okay, so there's nothing too complicated about it. Once again, the key thing to know is that they are complementary. Complementary means they follow base pairing rule. Now comes the RNA polymerase, the enzyme that is actually going to be making a copy of the DNA. So RNA polymerase comes. Are we trying to make a copy of the top strand, or are we trying to make a copy of the bottom strand? Think first. Okay. Don't answer impulsively. Are we trying to make a copy of the top or bottom strand? Think about that. Which one has the instruction to make the protein? Okay, let's take a look at the notes again here. On this picture, which one is the RNA? What color is the RNA? Purple, right? Okay. And the purple is same as what? Is it same as coding or same as template? The same as coding. Look, A, right? Do you understand? Right? Because the coding strand is the strand that has the instruction. The coding strand is the strand that has the codes for you to make the protein. That's why we try to make a copy of the coding strand. Okay? But in order to make a copy of the coding strand, you will be building from the template strand. That's what I'm trying to tell you, right? Remember with the coin experiment example, your mold has to be inverted from what you're trying to make. If the mold looks exactly like the coin you're trying to make, it's going to come out backwards, right? So if you want to build a coding strand, you have to be using the template strand. All right. So in our example over here, I have the template and I have the coding. What would the mRNA be? What's the first letter? Is the first letter A or U? U, that's right. It's going to be same as the coding. But because we're doing RNA, there are no thymines in RNA, only uracil, right? So that's why we have a U. Next letter will also be U. What's the next one? Is it C or G? It's the same, right? And you just copy from the coding. Do you guys understand? If you don't, please ask.
So why is it only the first two things that are used?
The first two T that becomes U?
Yeah.
Okay, because for the one of the differences between DNA and RNA is that there is no thymine in DNA in RNA, right? Okay, so anytime you see a T, you have to change it to. All right. So in the end, we have a strand that's identical to the coding. Identical, but what? They are identical in every single way, but that's right. T becomes U. Let's try one more example. Okay, so I have another template strand. And then I'm asking you to find the mRNA. For those of you who feel comfortable doing this, you can go directly from template to mRNA. If you are still a little bit unsure, then you can write the coding first. Okay, I'll give you a moment, help each other out. Hmm. Just the mRNA. That's you. What is the first letter? U. That's right. Because the relationship between template and mRNA is that they are, once again, what's the word I'm looking for? Complementary. Okay, so A pairs with U. Normally A pairs with T, but because we're doing RNA, it has to be a U. And the next one, that's right. And then you just follow the base pairing rule, right? We good? Yeah. Okay, one more example here. Okay, you try. Okay. If you see coding strand on the quiz, you should be very happy. Yeah. Why is that? Exactly, right? The relationship between coding and mRNA is that they're the same, right? Remember, the coding strand is the recipe. We want the recipe to be the same for the copy. So I just copy everything. But then if I see a T, I have to change it to U. You're confused. Here's the cheat sheet. Not that you can bring it to the test. What are the possible letters on the coding strand? A and then
B.
E, yeah. Sure. That's the only four possibilities, right? There's no other possible letters. But what about template? If it's ATPC on coding, what would it be on the template? E, A, right? Because they're complementary. What about mRNA? A, yeah. U. Why? Because mRNA is the same as coding, but P becomes U. And you have to understand this. If you get this first step wrong, the whole question will be wrong. Okay? And then the question is like 5 marks. For quick. Huh? The one question, one of these questions is 5 marks. If there are other things I have to teach. Okay? Don't take a picture, write it down. Take pictures of them, you're never gonna look at it again. So that's a summary. All right. Coding strands and mRNA are identical, except uracil replaces all the thymine because there are no thymine on RNA. You have to ask yourself consciously, am I writing DNA or RNA? You're writing RNA, there should be only uracil and no thymine. And then template and mRNA are complementary, basically what I told you. Okay, I need to know all of you got this one. Okay, don't yell at the answer, show me your finger. Okay, if you get this right, then I feel better, I guess. Okay, please think about it carefully. Yes, Rahafi, yeah. Anyone else? Look at the cheat sheet if you need to. Yes. Anyone else? Okay. This is template, right? It says right here, guys, templates, right? And what are we looking for? mRNA. Okay, good. So what's the answer? Good. Anyone else? Yes. Did you guys just not read it carefully? Was that the problem? Yeah, read the question 2 times, 3 times before answering. Okay. It's really scary. The people who are doing the online quiz, they spend only like, because I can tell how long they spend on each question. Some of them just spend 10 seconds on the question. I'm just like, they are answering instinctively. And they see the question, it's like, ah, it's B. Okay. Next one, it's C. Right? Like, don't do that. Okay. Read it carefully. Do we all understand why it is C? I hope so. Okay. I told you this yesterday, or like 2 days ago. Different version of a gene is called, huh? Good. Good. Anyone else? C. But for this one, what is this? That's right. Yeah. Try again. That's good. Anyone else? No, you were great the first time. Yeah. Different version of a gene is called allele. Okay. Allele. Allele. Okay. Let's take a quick note. This is called post-transcriptional modification. This is the pre-mRNA. It's the premature mRNA that we've just created by copying the DNA, by copying the gene, right? Okay. Initiation, elongation, termination. And it looks something like this. I'm not going to write the ACTG. I'm just going to use different colors here. Now, this is the 5' end, and this is the 3' end. Just like DNA, it has directionality to it. No, this is just got to be like a couple lines. Yeah. So it turns out there are sections on the pre-mRNA that are called exons. Okay. Exons are coding regions. You can think of exons are expressed in the protein. Exons are expressed in the protein. And then we have these non-coding regions, which are called introns. Right, so I'll call this like exon number 1, number 2, number 3, number 4. It turns out not everything that we've copied from the DNA is actually useful. Right? We've end up copying some non-coding material, some junk DNA, they say. Right? Think about, I don't know, as a loose analogy, it's like, you try to copy an article from a magazine. Okay, so you put it into the, it says coding region here, coding region. So it's like you try to copy an article from a magazine, right? Because you need it for your research paper. So you put it in the photocopier, you copy like 10 pages, and then afterwards, before you staple it, you realize, oh, I actually accidentally copy like an advertisement. Okay, it's not part of the article. I don't need it. So you remove it before you staple it, right? It's kind of like the introns are the advertisement. The introns are the fillers, the junk. That's not part of the code. Okay. So in post-transcriptional modification, step number 1 is called RNA splicing. And that involves the removal of introns and connecting exons. So afterwards, you are just going to have the exons. It's going to be shorter than before, but that's fine. This is 5 prime. This is 3 prime. And you have your exon number 1, exon number 2, exon number 3, and exon number 4. Okay. Now you might wonder, why would we have non-coding DNA? We don't have to know it, okay? Like they have functions, but we don't need to know. So basically, you cut out everything that's useless, and then you stitch these things together. It's kind of like making a movie. Sometimes you shoot the whole thing, and then you realize there is some useless stuff. You clip them, and you tape the rest together. Do you understand? There are like proteins that would help glue them together. What's the name of the protein? It's called the spliceosome. All right. We still have 2 more things to do for this mRNA here. If it goes out to the cytoplasm right now, it's going to get broken down very quickly, okay? Because the ends are actually not protected, and then it will just fray and disappear very quickly. And so we need to stabilize the premature mRNA by doing 2 things. Okay, so step number 2, add a special GCAP on the 5 prime end. Okay, so we put a special G right here. The G is just the guanine that we are used to, but it's modified guanine. Okay, so we just call it a GCAP. And number 3, we're going to add a poly A tail. Poly A tail on the 3 prime end. So poly A tail is basically a bunch of adenines. Okay, about 50 to 200 adenines on the 3 prime end. Longer poly A tail means it will last longer in cytoplasm. But remember, this is just a copy of the actual gene. You don't necessarily have to make this protein like forever. Right? In fact, sometimes you will want to make the protein just for the next hour, right? Or you want to make the protein for, maybe a day or 2. So the length of the poly A tail will help control how long this RNA is going to be in the cytoplasm. If the poly A tail is relatively short, then this thing will break down after some time, and then you won't make the protein anymore. But if you need to make the protein for the next couple of days, maybe, then you need to have a longer poly A tail. So you don't have to keep on doing the transcription over and over again. You understand that? So that's it. So after these post-transcriptional modifications, there are modifications that happens after transcription, right? That's why it's post-transcriptional. Then you have the mRNA. You have the mRNA. So just to recap with a diagram for you, guys, people are still writing up. Give you a moment. Any big test next week, by the way? No? And the week after? I should probably keep the quiz where it is. Okay. Over here, Tuesday. I'll see if I can finish. I hope I can. All right, so look, right? We talked about the DNA. We have 2 strands in the DNA. I want to ask you, don't guess, think about it. Which one? Which one is the coding strand? Based on the picture, is the top the coding strand or is the bottom the coding strand? Bottom is the coding strand. Anyone else think is the bottom? Anyone else thinks is the top? You're not supposed to see the letters, okay? Like just based on looking at this, you should be able to tell whether it's coding or template. Which one is coding? Top? Yeah. Okay, how do you know? Well, let's go back to this note here. When you actually build it, like when you're doing the question, it's different, right? But when you're actually building it in the body, you always use the template to make the RNA, right? Once again, you cannot use the coding to make the same copy because it will come out backwards. Okay? So based on this understanding, you should be able to tell that the top is the coding because the mRNA, the pink thing, is being built off of the bottom one. Okay, so coding on the top, template at the bottom. And once you build it, you can see that's the pre-mRNA, and it has exons, which is the darker pink, and then the introns, right? And then you would cut out which one do we cut out? Introns or exons? Introns, right? Okay, introns are insignificant. You can think of it like that, right? Cut them out. You put on the cap. What's the cap made of? G, right? And then poly A tail, and then it goes into the cytoplasm. All right. Okay, beginning of the gene is called what? Anyone else? No, not quite. Marks the beginning of a gene. Yeah, no. No. Yes. Okay, we wrote it down last time. Let me just show you one more time to help you see it, I guess. This is a gene. Beginning of the gene is called, everybody say it. No, promoter. Promoter, okay? And then the end of the gene, terminator. You know, like the movie Terminator? You stop, okay? Promoter, terminator. Okay, what is added to the 3' end? There you go. Yes. Okay, good, better. Yeah. We wrote it down literally just now, right? Okay. It is the A. Okay, guanine's at the 5. Okay. Don't think of as like, why do we have to know this? Okay. Just treat this as a learning exercise to remember things that seemingly is not useful. Because there's a lot of stuff you have to memorize when you move on to your next program. Okay. And not everything is necessarily directly applicable in life, right? I'm like teaching you how to learn, right? Basically. How about this? Okay. I'd be heartbroken if you don't know this. Okay. Very good. Good. Anyone else? That's good. Yeah. Anyone else? Yes. Yes. Okay. Introns, right? Okay. Because how do you remember it? Introns are insignificant. Okay. Exons are expressed. Okay. Now we're going to do translation. Just a little bit of review for you first. DNA and RNA are made up of nucleotides. We know that. And each nucleotide is either ACTG or ACUG. Nothing's new. Amino acids are the monomers for protein. And then there are 20 amino acids. Here's the thing. We know DNA codes for protein, but how on earth can you use 4 letters, ACTG, to code for 20 different amino acids? Because we have more things in the proteins than we have in the DNA. It turns out the code is not a single ACTG, it's not a single nucleotide. Even if you have 2 codes, is not enough. Right? If I have two letters, is not enough code. How many possible letters for the first blank? It could be A, C. Yeah, so how many letters?
4.
4. How many for the second spot?
4.
4. And 4 times 4 is only 16, right? How many amino acids do we have? 20. 16 is not enough, you see? And that's why we have to pop in a third one. 4 times 4 times 4 is 64. So this triplet, 3 letters, is called a codon. All right. Now we have this part you have to pay attention to, okay? Like it's very important that you understand this because everything that comes after this is dependent on what I'm about to explain to you. So DNA, this is template. I have to tell you, right? Remember? So A, C, C, whatever. And then when you do the mRNA, because it's template, A becomes U. We use base pairing, right? So now 3 letters, UGG is a codon. This codon codes for an amino acid, right? And the amino acid happens to be this thing called TRP. It's short form for tryptophan. You don't have to know the full name. You just have to write the abbreviation. Okay. And then the next code, UUU, codes for this one. And then the next code codes for that one, and so on and so forth, right? So when the body does translation, it is going to convert the mRNA into amino acids. And that's what I'm going to teach you how to do today. All right. The yellow sheet I gave you, can you take it out, please? Okay. On the yellow sheet, you are going to have all the codons, and I will always give you the codon chart on a test. Okay, you don't have to memorize it. All right, like some of you might think I'm crazy, but actually I'm not. So just like you won't have to memorize the periodic table, right? Now, having said that, even though you always have the codon chart with you, it would make it easier for you if you remember a few of the codes. For example, okay, just backtrack a little bit first. I want to ask you again, what do we call the beginning of a gene? Promoter, thank you. Okay, and what do we call the end of gene? Terminator. Similarly, when we build a protein, we need to have a start and we need to have an end. Okay, the starting codon is going to be AUG. Right? So remember, August is like beginning of the school year, start of a new school year, or something like that. Okay, so AUG is always going to be at the first amino acid when you build the protein, and the amino acid it codes for is methionine. Okay, MET. Now some people ask, does that mean you cannot have methionine in the middle of the protein? Not necessarily. Okay, you start with it for sure, and maybe along the way you're gonna have it as well. Think of writing English sentences, okay? The first letter of the first word is always, no, like when you write a sentence.
Oh.
When you write an English sentence, the first letter has to be capitalized. Thank you. Does that mean you cannot have capital within the sentence? No, right? Like if you have a name or whatever, right? You understand? Okay, so like MET is like that capital letter. It always shows up at the beginning, okay? So I want you to remember, start codon is AUG. Got it? When you come to a stop, there are 3 stop codon, UAA, UAG, and UGA. I want you to remember it because not for this quiz, but like for later on when we learn something else, it would be helpful if you memorize the stop codon. You can do the question much quicker. I have a cheesy way for you to remember this, okay? So you got A in biology. You are awesome. You get it? You are awesome. And then you are good. Okay. They're very cheesy. If you goosebumps, but it works, okay? So stop codons. Understand? What's the start codon without looking? No, start. Start is AUG, guys. Start is August, okay? Remember, start is August, and then end is one of those things. The beginning, end, okay? Got it?
AUG is the beginning.
AUG is the beginning. August is the beginning of every school year, I said. But it doesn't matter. I'm just making stuff up, okay? Like whatever works for you guys. Of each protein. Protein, yeah. It's always gonna be that. Okay. All right, so here we go. We're gonna totally do this. Okay, you got this. It's very easy. You'll love it. Maybe. All right, this is a different chart. Let me just You're over first. Okay, so take out the yellow sheet. I'm going to teach you how to use it. All right. So here we have, yeah, get one if you don't have one. Okay, so yellow sheet on the side. Right here, it says find the amino acid sequence encoded by the following mRNA strand. I already transcribed it for you, right? Okay, so let's take a look here. The first one you see is AUG. Without even looking, you should know AUG is MET, right? That's the amino acid it codes from. Right, you don't need to know the full name. I just like saying them because it's easier to say. Okay, how do you look up AUU? Okay, so A, this is first letter, so I have to find where is A. So this is U, this is C, this is A. So it means it's gonna be this row here. That's the first letter. You understand? You look for the first letter at the first letter, okay? And then you're gonna look for the second letter, U, which is up here, right? And the second letter is U, which is right here. So then you don't have to highlight it, but I will. Yeah. So that means the second code is the intersection of the row and the column, which is right here, right? And I'm looking for AUU. AUU is ILE. Okay. A good job for telling this is ILE. Lots of people think it's IIE or LLE. Okay. But if you're able to tell difference, really good. It's ILE, it's isoleucine. There you go. Okay. Let's do one more in case you don't get it. We're looking for CCU. Okay, watch if you don't get it. C, I look for C, right? C is right here. And then what's the second letter? Which is right here. So it's in this box, right? And what is CCU? Pro. So easy, okay? Do the rest of it. If you don't understand, that's okay. Just ask me to come over, okay? If you need help, let me know.
When it says stop.
And you write stop.
Oh,
You're
like
on
that's
the
like the end.
Yeah, that's the end of the protein.
I think.
Too small or something at the back. Hey, can someone yell out CAU for me, please? His, that's right. His, HIS, history. Anyone need help? I can come and help you. No?
What is this word?
They are amino acids. Remember? We build the amino acids together, like the little beads for the necklace.
Oh,
Yeah, 20 amino acids, right? Okay, CGC, guys. Arginine, yeah, and then? Yeah, TYR, and then? What is it? PHG, yeah. PHG again. And then UAA is you are awesome, which means stop. Okay, listen. You have to put the word stop so that I know you know to stop. Okay, if you leave it blank, I cannot give you marks. Right? It's a balancing equation, right? Even if it's one, you have to put one, right? Same thing. Get it? You guys will get the 5 marks. So excited. Okay, 5 marks for the whole quiz is like I don't know, almost 10%. Right, almost, but not quite. When's the quiz? Quiz is Tuesday. Okay, show me all of you can do this. Great. Don't yell it out. Okay, just show me your finger. You have the chart in front of you. You don't have to squint. You have the yellow chart. You have the chart.
Oh.
Okay, good. Good. I don't know. Oh, yeah, good. Okay, good. Yeah, you got it? Good. Yes. All right. At least half the class is here, and at least I know half the class can do it. Okay. The other half, I don't know. You guys teach them, okay? I know. It's like nice weather, and some people might be. Why don't you what? Sure, sure. Should be there. Okay, I'm gonna skip this question. It's probably too hard at this moment. We'll do that later. I do want you to try another one here. Okay. So it's a little bit more tricky now. Okay, a little bit more tricky because, oh, you know what? This is the same one. Okay, we don't have to do this. I think you already know that. I might have copied the wrong one. Oh, there we go. Example 2. How do I do this? Okay, now this is a little bit more tricky because I did not give you the mRNA, right? What is this strand? What does it say on the screen?
Stop.
No, this strand. What is this?
Template.
Template, right? So you have to read it, right? Template strand, and now you're gonna make your own mRNA. That's why you needed to understand this. Because listen, if your mRNA is wrong, your protein will also be wrong, and then I cannot give you marks. Okay? So do it. Go, go, go. Use the yellow sheet, guys. Okay, don't squint your eyes. Okay, in one word, tell me what's the relationship between template and mRNA? No, the relationship between template and mRNA. Complementary, right? Complementary. So that means this one is going to be A, U, G, and then so on and so forth. Okay. Now you check my work because sometimes I do them wrong. Now, if you easily get confused because you have messy handwriting like me, you might want to put like a bar to separate them. Okay. So, you don't lose track of what it is. Is it okay? Manuel, you get this? Yeah. Just check it. And for the protein? I have no idea what the right answer is. I'm just copying what they say. Can you please verify? Yep. Which one? Which one? This one?
LEU.
Is it? Let's see. UUA. UUA is right here. Yeah, that's LU. Everybody okay? No? Okay, I'll come help you. But let's try another one. Right, go ahead.
I'm so sorry. No, no, no. I think I'm okay.
Not always, depends what's given to you, right? So if I give you the template,
Yeah.
then A pairs with you. Is it?
Okay, because I'm just confused, because on here, I was like, okay, it's supposed to copy the coding.
That's right.
mRNA, right?
Exactly.
But then why is the one that's like the mRNA a T, why is it A not a U?
Why is the A not a,
Yeah,
are you reading the A from the coding or are you reading
from the coding.
Yeah, okay, so A is an A because coding and mRNA are the same.
Okay, wait, so mRNA copies from template?
mRNA copies coding. When I say copy, I mean identical.
Oh.
So because this one is coding,
Yeah.
the coding, just copy all the letters exactly as it is, but you have to switch the T to the U. That's all you have to do. Switch the T to the U if I give you coding.
But coding is different.
And template is the base pairing. Okay. Got it? All right. So the key thing to know here is that this is coding, right? If it's coding, then your mRNA should be the same. But the T becomes U. All right, for the protein, here we go. MET. What is it? Oh, THR, yeah. And then GCA is ALA, right? GGC, ACU. So don't freak out if the last one is not a stop, right? Because in real life, the protein can be very long. Some proteins are hundreds of amino acid long, right? So maybe this is going to give us hope. Right? Similarly, if the first one is not MET, like again, doesn't mean you're wrong. It may be it's like in the middle of a sequence, right? It's not always like at the beginning. Are we okay? All right. So I have to teach you one more thing. This is the last thing on the quiz. And then I'm gonna give you a worksheet and you have to finish the worksheet with the correct answers before you can leave. Okay. I'm locking the doors. I said I'm locking the doors until you get the right answer. Okay, actually, how about this? If the codon contains 4 nucleotides instead of 3, how many amino acids can we code? Very good. A lot of you got it. Anyone else want to try before I tell you the answer? So if we have 4 nucleotides instead, then it's 4 times 4 times 4 times 4, right? So 64 times 4 is 256. All right, we're gonna do translation quickly. So for translation, we need to have 3 kinds of messenger. We have to have 3 kinds of RNA. And we already know messenger RNA. Messenger RNA is the copy of the gene. It's the recipe, right? The recipe. It tells you what amino acids to connect together. In fact, we just learned how to read those codes. But we also need additional RNA to help with the process. The rRNA is going to be needed to build the protein. Okay. The RNA, if you remember, it's what makes up the ribosome. So this is the recipe. This is like the chef that will be cooking the recipe. And then we have the tRNA, which stands for transfer RNA, which will be bringing the correct amino acid to the ribosome. And so that's like a little helper that will bring the ingredient. Recipe, actual making of the protein. This one brings the amino acid. So I'm going to give you a note. And this is the last thing that you have to learn for the test, for the quiz. Okay, so the tRNA looks like this. First of all, it's RNA. So is RNA single strand or double strand? RNA, yeah. Single strand, right? Okay, so a single strand like this, but this single strand actually can loop back on itself and form a structure. I don't have my props here today, okay? But it's like, it's like this thing, okay? Like it's able to loop back on itself and form these hoops, okay? So in fact, we have the tRNA here. It's gonna fold back on itself like this. Okay, it's gonna fold back on itself and form this like clover leaf. And over here, for example, let's say this is ACGG, and then the other side would be UGCC. Right? It just happens to be complementary, and then they will be holding the shape in place. Okay, so similarly, they will hold each other in place like that. And it forms this clover leaf structure. Okay, so this is complementary nucleotides. Now over here, this is going to be the amino acid binding site. Uh, oh, this thing is the amino acid. It's gonna be carrying. You know the
But is there, like, do I leave that hole here?
oh, it's not a hole. It's not a hole. It's like a ball.
No, I'm saying like,
What?
in between, like, where the.
Oh, this hole?
Yeah, that.
Oh, yeah.
Leave that open?
Yes, please.
Oh, okay.
All right, misunderstood. Now, very important is I mean the whole thing is like ACUG, right? Okay. But at the bottom here, there is three nucleotides that are especially important. Okay. So this one is like, let's say is ACA. I'll explain what it means. This is called the anticodon. It's located at the bottom of this tRNA. Okay. And the anticodon is complementary. Okay. This is about to get more confusing. It's complementary to the codon. I just write down and I'll try to explain. Now remember earlier we had the mRNA, right? This is the mRNA. We already cut out all the introns. That's why it's only one color. And then the mRNA is just a bunch of ACUG as well, right? And on the mRNA, every three nucleotides is a codon, which would tell you what amino acid you need to bring over, right? So let's say the mRNA is like a bunch of ACUG, but at this specific spot, let's say is UGU. So UGU, what amino acid are we looking for? CYS, right? Okay. So how does the tRNA know what's the right amino acid to bring? Well, let's say this is PHE, okay? PHE, you just have to believe me for now. Don't look it up because you'll be looking it up wrong, okay? PHE would have an anticodon of AAA. So when this amino acid comes, take a look, please. Heads up. When this amino acid comes over here and try to bind with UGU, is it going to fit? No, because AAA is not complementary to UGU, right? So this one try to fit, but it's like the wrong key. It's just gonna bounce off. Right? But this one comes over and ACA is complementary to UGU, right? And this one is actually going to be carrying the correct amino acid. Okay? So again, every tRNA will have its own amino acid, but whether it fits or not has to do with whether the anticodon is complementary to the codon. Okay? I copy this down first. I will give you an example to work through together. Okay, please.
Like the line. No, like scroll up. Like that's T.
Yeah, but then that's like not folded up yet, but then it will fold up
this
into
is like-
a key. Yeah. You have a question? No, ask. I'm sure other people have the same question as well. No? Okay. Okay, don't copy this down. Okay, I have to work it out. I'm making it up on the spot, and I might get it wrong. Okay, hold on. Okay, this you can copy. Yeah, this you can copy. It's fine. Yeah.
So tRNA has this anticodon here. So it's the when we transfer it into the codon, C goes to G,
Good. Yeah,
U goes to A,
that's right. Yeah, yeah, yeah.
U goes to A.
Yeah.
So this is amino acid.
This is based on this.
Yeah,
Exactly.
But you, your slides put instead of this one.
Which one is it? Which slide?
In your slides, if you go to your slides,
This slide?
yeah, I'll show you.
Oh, you mean like when I was doing it? Which one? I didn't check. Is it this one? CCG? CGC.
After this, you have questions in the TRN section. You have questions. You will find it over there. Instead of this one, you put this one as an answer.
Can you show me the slide afterwards?
I'll show you. Yeah. I can bring you my computer. I'll show you.
I'll come. Okay, let me explain this first. Okay. All right. So here we go. Translation.
The other one is TRN.
Yes, this is translation. That was just tRNA, like in general, like yeah. A little bit. Okay, but this next part don't copy because I'm gonna be moving stuff around. Okay. Okay, so I'm gonna have like AAG here. And I'm gonna have UAC here. And then let's say this one is UUC. Please pay attention to this next part. What do we call this thing again? Anticodon. What do we call the whole thing?
Yeah.
tRNA, right? Okay. This is an amino acid, right? What would this amino acid be? If the anticodon is AAG, what is the amino acid? Please think about it. All right.
Oh, no PhD.
PHG? No. Anyone else?
Nice.
LYS? Okay, anyone else? Who's it? LYS? Okay, who thinks it's LYS? Only 3 people? 4 people? 5 people? Who thinks it's PHE? What about the rest of you? Okay, listen. If on the test or quiz at any moment you find yourself looking up an anticodon on the yellow sheet, you are doing it wrong. Okay? You never look up the anticodon. What do we call this chart? The codon chart.
Wait, sorry. Okay.
Well, this is tRNA, right? On the tRNA is an anticodon, right? So listen, what should you be looking up instead? Well, if the anticodon is AAG, what would be the codon that it binds to? What's the codon? UUC, right? So please look up UUC. What do we get? PHE, there you go. Okay, how about this one? Go ahead, look for it. What is it? So again, we do not look up UAC, right? What do we look up instead? There's no T, remember? AUG. What's AUG? Matt. And then the next one? Yeah, what's AAG? LYS, that's right. Okay. So that's what happened. This is the ribosome, and the ribosome actually has like 3 slots in it, okay? I'm not going to draw it, it's too confusing. There's 3 slots in it. Okay? And you don't have to draw this because I'm going to be moving things. But watch. We have the ribosome, we have the mRNA, and then let's say we want to build the first amino acid, right? So this guy's going to come and he's trying to fit in. Is it going to fit? No, it's not going to fit, so it's going to move away, okay? And then, another one is going to try. Is this going to fit? Yes, that is totally going to fit, right? So it's going to stay there. And then we're going to try the next one, which is like, what is this? UUC. That's right. So AG. So look, this is going to come. It's going to come over here. And then the MET will break off. And then you will form a peptide bond. And now it's going to be on the PHE, right? And then this guy, I don't know how I'm going to do this. I'll try. It's not going to work. This guy is going to fall away. There you go. Okay. Leaving behind a little bit of stuff. Okay. And this is a ribosome. And then this is empty now, right? So it's going to have to find another amino acid. Right? It's going to have to find another MET. Right? And then it's going to bring it to another ribosome. Do you understand? And then the next one is going to be, I think it's this one, right? It's going to come. And then same thing. Watch. The growing one will break off. And then it will go to the new one. And then this one is going to fly away. And then now it's going to reload. So far, so good. Then the ribosome will move forward. You don't have to draw this yet. It's impossible. The ribosome is going to move forward. And then we're going to wait for the next one. Okay? What's the anticodon of the next one? DAU, that's right. And so on and so forth. Until we get to the stop codon, there are no transfer RNA for stop codon, and the whole thing just stops and falls apart, and then you will release the protein. Okay? Yeah, go ahead.
Anticodon, right?
Yes.
How about mRNA?
mRNA is codon.
Yeah.
Yeah. RNA is just a ribosome. It builds. There's no code
So
on it.
it's not-
Yeah. Mhm. Okay, so here we go. Let's try one more time. Okay, one more time, please. First of all, we have these mtRNA. Okay, they have to load up with the amino acid, right? They got to pick up the cargo. Right. So this is GAG, anticodon. Which amino acid should it pick up? Look it up. Right? Because we look up CUC. Okay, and then this one's gonna load up with that, right? Okay. And then once they load up, this is basically what I'm trying to tell you. Right? The new one will come, and then the growing strand will break off and attach to the new one. The empty one will leave and then go pick up another amino acid. And then it just keeps on going and going until it gets to the stop codon, and then everything gets released. Okay, everything gets released. All right, that's the cutoff for the quiz on Tuesday. Okay, I'll post a review video for you Right now, I want you to do this worksheet. Okay. Oh. So now we're gonna add the anti-codon here. Anti-codon. What letter should go here? Is it U or A or T or what are we gonna? U. Uh-huh. All right, work on the sheet. I'll come check it. You can leave when you get it right. Yeah.